Hello, everybody, and welcome to our first virtual frameworks event. We're going to have Lori Leroy from BioCrossroads kick us off. And just wanted to let everyone know all the attendees are on mute. So we would like for you to use the Q&A tool on the webinar to send any questions to the panelists. We'll do our best to get to answers today. And if not, we will definitely address them and get them out to the group once the webinar is over. And with that, I'll pass it off to Lori Leroy, Vice President at BioCrossroads.
Welcome, everyone, and good morning. Glad you could join us for our first virtual frameworks. We're really excited to bring this content to you today, and hopefully you can eat your lunch and learn some really great information about work that's happening here in Indiana. First of all, I want to welcome everybody and thank our sponsors, Indy Science Connect and the Indiana Biosciences Research Institute. Today, we're talking to you about the impact that COVID-19 has had on point-of-care diagnostics. To say our lives have been completely disrupted is an understatement. COVID-19 has changed the way we work, live, and play. It's also changing the way that we conduct research and development, and especially with new technologies. Point-of-care testing has become significantly more sophisticated over the years, meeting clinical needs for quick and accurate test results where it's needed and when it's needed, as we all know all too well right now. But having available and accurate testing in minutes isn't enough. We have to be able to test the masses and test them quickly. Point-of-care testing has never been more important than it is today, and the entire world is now watching and waiting to see how the industry responds. So with that, I would like to introduce our first speaker, Dr. Michael Pugia, investigator and director of bioanalytical technologies at the Indiana Biosciences Research Institute. Dr. Pugia works to develop new single-cell bioanalytical technology for measuring proteomic biomarkers as targeted diagnostics for the fields of endocrinology, oncology, and infectious disease. Michael has spent 30 years in the biomedical in vitro diagnostic industry for Bayer and then for Siemens Diagnostics divisions. So please welcome him, and we look forward to hearing what he has to say today.
Okay, so thank you. Hopefully I can advance the slide. There we go. Okay. Wrong way. Sorry. Okay. So I'm just going to jump into the presentation a little bit. I guess it sort of overcorrects a little bit. So really, point of care technology, got its start in Indiana. And I don't think a lot of people really know that, the urine test strips and the first glucose test strips were invented here in the late 40s and 50s. And they had pretty much evolved to a mature product line by the 80s. A lot of this work was led by Alan Helen Free. And in the 80s, there was really a paradigm change to go from optical dipsticks to electrochemical sensors. And today, those are still, manufactured here by Roche. These companies, developed the idea of a glucose meter. I mean, they actually coined glucose meters and the word strips. So when I left, or when I started here in my career in the late 80s, point of care testing was still, pretty hot topic. But it really stalled in the 90s, and really it was due to a much needed regulation of the whole field, which was the CLIA Act. And then the cost of capital to bring in high volume manufacturing really stalled it a lot. But really, I think the final nail on the coffin has been the cost that it really takes to validate and verify a clinical diagnostic marker. I mean, few people know that, literally maybe one test a year, one new marker a year makes it to market. But with that said, I was, able to be exposed to, the leading edge people who were at a company called Miles or Ames division at the time. And one of those was Dr. Lowry Messenger. And he really developed a state-of-the-art test called the Diabetes Care Analyzer. And it was able to really set a standard of quantitation for a point-of-care immunoassay. Fortunately, he picked a really good one to do, which was hemoglobin A1C, and they brought that to the market, just one test. And now that's sort of an everyday word for everybody who's a diabetic. They know what hemoglobin A1C is, but it's a real challenge to bring something like that to market. And it was really a tribute to that team to do that. At the same time, I was on another team led by Dr. David Ledden, who thought, well, measuring one thing isn't going to work. We need to measure many things. So they took the pregnancy testing concept, which had just been developed, immunochromatography, and brought that to a strip reader and developed the whole concept of optical imaging. Long before we had cell phones and even CCD cameras. And then, one of the markets that they focused on obviously was, infectious disease, the flu test. But these technologies had limitations. They were yes/no tests. And really, a lot of point of care devices have not really helped this whole thing. A lot of times they lack cost effectiveness. So it's just easier to get an accurate answer from the lab the next day. And rarely do they both achieve a high clinical sensitivity and a high clinical specificity. So somehow this concept of allowing, 50% false positive rate or a 50% false negative rate became acceptable. And it's really caused a lot of issues. One of the biggest issues it causes is antimicrobial resistance. So in the 2000s, actually at 2000, I was privileged to be on some really leading edge lab on a chip teams. One led by with a company called Microparts. Gert Blankenstein, was doing the first microfluidic designs for lab on the chip. And then later with Dr. Walter Gumbrick, who had literally, fathered the lab on the chip design. This shows those devices. And we really had the hope that miniaturization and taking these designs was going to allow us to, reach more markets, more assays and stuff like that. The issue is we ran into a lot of laws of nature that we hadn't considered, and the cost was really, the cost of even the device and of validation and capital were just enormous to do a technology change. In the 2010s, a lot of the microfluidic technologies did survive. I was on a project with Walter Gumbrick where we were able to take them into the central laboratory and do more sophisticated tests that you would charge more money for. And one of those was circulating tumor cells, where we were able to isolate a handful of cells out of a tube of blood and determine if you still had circulating cancer cells during metastatic cancer. The issue with that is, it takes 3 or 4 days. It's only done in the lab. Only a very small number of people get those kinds of tests. They're very helpful for directing chemotherapy. At the same time, our interest in industry started to tend towards mass spectrometry. And I don't know if few people probably know that, Indiana is the epicenter of analytical chemistry, like the leading state for analytical chemistry. And one of those key fields is mass spectrometry. And these guys work together well across all the universities. They recently formed a National Science Foundation center called the Center for Biometrology, where they're collaborating in this space with industry. One of the key people in the field who literally fathered mass spectrometry is named Grant Parks, and he had invented at Purdue a miniature mass spectrometer. So we thought, well, maybe we could try that to try to get to a point of care device. Again, a very high tech approach to try to achieve it. At about that point, I became a little frustrated with the industrial approach. I mean, Zoom meetings and industrial collaboration, we've been doing this for like 20 years. All of our teams are all over the place. We thought, well, why don't we just try to focus it a little bit more on a collaborative model where we had more boots on the ground in more places where we could actually do more hands-on work and try a collaborative model across the ecosystem of Indiana by taking industrial scientists and putting them into the Indiana Biosciences Research Institute and seeing if we could try some collaborative models. So this project that I'm going to share here and some of the speakers today are part of that team effort. And this was the approach that we took, which was really not to start with technology, but to start with customer needs. And we did that with a very cool market analysis system out of Notre Dame in the Mendoza School of Business, which was the STEAM program. And they basically take a really smart student and they give them access to all these really cool Notre Dame alumni, many of them who are doctors, and get to ask them a series of questions and keep peeling back the onion until you can get to the real heart of what the design needs to be. Once we had that idea, all the universities really need to have these core high-tech facilities. And the one that we were working with to get the initial core technology done is the NanoFab facility that Dr. Patrick Fay has at Notre Dame. And so we can make some of these sensor concepts to try to address the market need. He rightly, directed us to a technology provider, which is Indiana Integrated Circuits. It's a startup up in the Notre Dame ecospace, and Mr. Jason Kulik is an electrical engineer who got his degree at Notre Dame and decided to become a technology provider. And by that is somebody who helps do the tech transfer, take the design out of the NanoFab facility and get it into the hands of people who are going to license it. And he's extremely gifted at doing that. So once you have the core technology, you still need to create a device around it. So it wasn't too hard to find, one of the leaders in biomems, which is another term for microfluidic integration, Professor Weirley. And we did a collaborative project with his PhD candidate student, KZ Gupta, and simplified the design from a big giant robot to a little handheld. So that's more of how the device works, but to really get it to work right, you need really sound analytical chemistry direction, especially in this domain. It was in electrochemistry, so we didn't have too far to look to find Frederic in analytical chemistry, an up-and-coming professor at IUPUI, and she was very, very gracious in sharing 2 undergraduate students that became a fixture, and to help really direct some of the industrial postdocs that we had, which really needed more of a professor's touch. So even with that, if you get all that done, you still need access to the medical school. And we were blessed. The application that we set out to do was pathogens that were resistant, biothreats. We weren't thinking about COVID. There are actually worse ones out there. And we were thinking about bacteria, film-forming bacteria. And fortunately, Professor Sen and Dr. Roy at the IU School of Medicine and the Department of Surgery are like the world's experts in this field. And they could get us straight on what we needed to do to test to get us these early samples. And also, at the same time, we have a great pathology department with key opinion leaders. Gerald Denny's is one of those in microbiology and gave us access to samples for testing. So that's a long-winded description of how it worked. Hopefully I'm not too far off track on time. So once what the team tried to do was change the paradigm. And a lot of this is just direction that you get from grants, from the NIH, stuff like that. They want to be able to take a complex sample, a milliliter, and process it immediately, and isolate anywhere from down to about 10 pathogens. I say virus here, but any kind of pathogen, and be able to detect and enumerate them and quantitate them, and to do that in anywhere from 1 to 30 minutes, and to do that in a highly multiplex panel. The panel concept that we did was 341 results. And it would process anything, any kind of complex sample, blood, urine, so you don't have to keep changing it. And it'll do a microliter up to a milliliter. So, but the concept that really came across was, let's not have to get another sample. Let's take that sample after it's tested and send it to the laboratory to be confirmed with even a higher multiplexing biomarker platform that's much faster than what's out there. And that, of course, is the mass spectrometry, which came out of the collaboration with Professor Cooks on a technology called Sierra technology. And that really allows 28,000 results in a day, which is an order of magnitude greater than the current platforms. At the same time, we knew that it was essential to preserve the genetic information, so the same sample can have the genetic information. And as long as we were capturing the cells, we wanted to show that we could clean the cells and release their DNA and RNA, and be able to just use regular PCR to identify the actual mutations in the pathogens. And so I'm happy to say this is, pretty much been all written up in a paper. Just to look at the technology, core technology a little bit closer, this is the actual sensor. It's the size of an ELISA well. It has 341 micro wells in it. Each micro well is a separate sensor. At the bottom of it, it has a slotted great electrode that allows capture of only what we want to detect and passing away all the other interferences. Sort of the magic of Jason's design was to put a little electrode in each one. And one of the easy benefits was to show that we could self-array individual capture beads easily during manufacturing into all these wells, so that we could get 341 addressable answers. And not really, once that's done, the mass spec reagents can be just released with a simple acid, and then they're run on the mass spectrometry, or we can lyse the captured cells and do their DNA and RNA. So it all comes down to laws of nature and not trying to break laws of nature. So there's really 3 working principles of any kind of complex point of care system. One is isolation. So you're starting with a complex sample and you want to get it down to just the rare molecules that matter and into a small space rather quickly. This comes down to things like porosity, materials, pore sizes, stuff like that. The other thing you want to do is you want to be super sensitive, more sensitive than anything that's out there. Currently, immunoassays have a sensitivity floor of about 0.1 nanomolar. Sometimes they can get a little bit deeper into the picomolar. But by using the microwells, we're able to concentrate all that signal into a smaller volume, and that's allowed us to hit 6.6 femtomolar is what the last test results showed. So that means that that's significantly more sensitive than what is out in the laboratory right now. And that allows us to use less samples, less costly reagents, or to detect things a lot earlier. At the same time, multiplexing is key. I mean, we all know that, multiple sources of data just make it a lot better. So we wanted to keep optical, which we managed to. We wanted to use spatial, which microwells, like I showed, 341 microwells. And then we wanted to go to barcoding, which is a concept, mass label concept that was developed that allows really unlimited, each antibody can have a different barcode. We weren't really working on COVID. It wasn't one of the key pathogens to worry about in February. I mean, we were aware of it, but today we're working on it. And so I think COVID is a good example of trying to get actionable information quickly. So there's really a couple of different things that need to be measured at the same time. One is, do we have live virus? Do you have dead virus? And the more sensitive you are, the further down in the incubation window you can detect. So if you can get closer to 0 days, you're able to contain people quicker, right? So, and if you can do that within a few minutes, you know that those people have an issue and that they could be properly treated. That's called actionable information. So with 6,000 point of care systems, you should be able to screen 1 million people in a day and still be able to take the collected virus and send it to the pathology department to do a deeper dive. Maybe the resistance has changed, maybe it's mutated. The other thing that really is needed is to tell how a person's immune system is reacting. And the immune system really reacts in about 7 days by producing a molecule called IgM. So you have to be able to detect that first. That IgM response goes down in about 21 days, and then our more permanent response, which is our antibodies called IgG, are there. So you can see that you need a multiplex of different results to really gauge where a patient is in the work process. And we need to think beyond just one particular virus, right? This might be, we want to be able to detect more than just one thing at a time. We also would like to know how the patient is responding in his immune system. People have different abilities to respond to infection. So that is everything I had to share.